Good morning, everyone. Uh, my presentation topic is reverse phase HPLC analytical method development and validation for simultaneous estimation of Allopurinol and Lesinurad in bulk and its tablet dosage form. So, what is that RP-HPLC means? It's a reverse phase high performance liquid chromatography. It's a separation technique which is used to separate the components from the mixture. So I developed this combination allopurinol and Lesinurad and drug combination. I developed that analytical method, and also I validated that method as per ICH guidelines. And also I applied that stability studies for that and developed analytical method. Myself, Dr. Ismail, Associate Professor, Crescent School of Pharmacy, BS Abdul Rahman, Crescent Institute of Science and Technology. So this allopurinol and Lesinurad, which is used for gout. It's a chronic inflammatory arthritis that affects adults. It is marked by a greater level of uric acid in the blood. Increased level of uric acid, so normal uric acid level is 2.5 to 7.2 milligrams per deciliter. So the increased level of uric acid in blood is called gout arthritis. So my selected combination which is used to treat that gout arthritis. So this allopurinol, which is act as a xanthine oxidase inhibitor because it is an enzyme which is produce that uric acid. So that enzyme is inhibited means uric acid production will be stopped. So allopurinol and Lesinurad combination is selected for this analytical method development. This is a chemical structure of allopurinol, and this Lesinurad also has to decrease the uric acid level. So it is a combination dosage form. So it's a structure of allopurinol as well as that Lesinurad. However, the problem statement is the existing LC methods were less convenient and time consuming, which were not suitable for individual and simultaneous estimation. Simultaneous estimation means at a time we can determine more than one drug. So this selected combination, which has two drugs, one is allopurinol and Lesinurad. The HPLC system reduces the time and significant cost of analyzing samples with better result, because by using HPLC we can complete the process within the less time, because in industry we have to do many samples. So the time reduction is very important, as well as the cost reduction also is very important. High performance liquid chromatography allows an analyst to work on superior skills with much wide range of linear speed, solvent flow rates and speed back pressure than traditional HPLC. Considering the growing demand for the above titled drug in global market, it is necessary to develop a new economic, accurate and rapid HPLC analytical method for simultaneous estimation of allopurinol and Lesinurad in pharmaceutical formulation, and to conduct force degradation studies. What is the need of these force degradation studies? Means the drug is stable in which condition we can find out by using force degradation studies. Many force degradation studies are possible: acidic, alkali, oxidative, photolytic, thermolytic, like that. So we have to carry out in this condition, and we have to find out which condition the drug is stable or unstable. So the problem statement is the pharmaceutical industry is under intense pressure, increase productivity and release new market drugs into the market. Analytical chemists are challenged to find faster ways of delivering quality data across a range of project needs. A number of approaches are being employed to increase separation throughput. A new tool to meet these challenges became available, and this new class of separation science is known as high performance liquid chromatography. The need for speed has been driven due to large number of samples that come particularly in drug discovery and analytical laboratories. HPLC gives good resolution. The resolution means the separation between the two components, speed and sensitivity, and this is achieved through a use of small columns. Because HPLC we are using stationary phase and mobile phase. The stationary phase is called as column. It is made up of stainless steel material which is packed with packing material like silica, aluminum, like that. In the present scenario, HPLC has better scope. HPLC is an emerging liquid chromatography technique which enables significant reduction in runtime and solvent consumption. The research work embedded in this dissertation is to develop stability indicating analytical method and validation for the simultaneous estimation of allopurinol and Lesinurad in bulk and its dosage form. So we can use this method for pure drug and as well as for different dosage form by HPLC technique. So the aim of this research is to develop reverse phase HPLC method for the simultaneous estimation of allopurinol and lesinurad in bulk and in dosage form, and we have to validate. Objective literature survey reveals that there are no analytical methods available for simultaneous estimation of allopurinol and lesinurad in bulk and in dosage form by RP-HPLC technique. Hence, our present plan is to develop new, simple, precise, accurate HPLC analytical method development and validation for the simultaneous estimation of allopurinol and lesinurad in bulk and in its dosage form. So these are the chemicals used: allopurinol, lesinurad, potassium dihydrogen, phosphate, water, and methanol. So always, what are the chemicals we have to use for HPLC? We have to go for that HPLC grade solvents only, and acetonitrile, water, orthophosphoric acids. So these are the instruments I used: HPLC, UV visible spectrophotometer to find out the overlay spectra, pH meter to find out the pH, weighing machine, pipettes, and burettes and beakers. So I did many trials. The first trial with the chromosyl C18 column 250 mm, 4.6 mm, 5 micron particle size. The mobile phase was sodium phosphate buffer methanol 50-50 ratio. The flow rate is 1 ml per minute. The wavelength is the lambda max 255 nanometer. The injection volume is 20 microliter. So we didn't got proper peak shape for these two drugs. The second trial with the same chromosyl C18 column 250 mm into 4.6 mm, 5 micron particle size, water into acetonitrile 30:70, volume by volume, flow rate 1 ml per minute, wavelength 255 nanometer, injection 120 microliter, and peak obtained here, that one also somewhat broad in shape. Trial 3, same chromosyl C18 column 250 mm into 4.6 mm, 5 micron particle size, phosphate buffer, methanol, pH 2.5, 30:70, volume by volume, flow rate 1 ml per minute, wavelength 255 nanometer, injection volume 20 microliter. The separation of two analytical peaks was not proper, so the mobile phase ratio has been changed for next trial. Trial 4, the same column, but the mobile phase is phosphate buffer and methanol, pH 2.5, 20:80, volume by volume, flow rate 1 ml per minute, wavelength 255 nanometer, injection volume 20 micrometer, and this one also peak shape is not proper. The last one, trial 5, so I used a chromosyl C18, phosphate buffer methanol, 10:90 ratio, and this one also peak shape is not proper. So the trial 6 with the same column, chromosyl C18 column 250 mm into 4.6 mm, 5 micron. The buffer is phosphate buffer is methanol, pH 3.5, 55:45. So we got somewhat a better peak, but the fronting occurs in that peak. So it is due to saturation of stationary phase. So we go for next trials. So finally, this chromosyl C18 column 250 mm, 4.6 mm, 5 micron, phosphate buffer acetonitrile 70:30 ratio, flow rate 1 ml, wavelength 255, injection volume 20 microliter. So here the peak shape was good, and the plate count was 200, tailing factor below 2, resolution is above 2 as per ICC guidelines. So we can take this method as an optimized method. So after this many trials, the optimized chromatographic conditions are: the instrument what I used is water HPLC with the auto sampler and PD detector, temperature 25 degrees Celsius, mode of separation isocratic. Isocratic means throughout the separation we are using same polarity of the solvent. Column Inertsil ODS 150 into 4.6, 5 micrometer, buffer phosphate buffer pH 3, mobile phase phosphate buffer pH 3 is to acetonitrile 70:30. Flow rate 1.5 ml per minute, wavelength 255 nanometer, injection volume 20 microliter. So these are the chromatogram I got for the standard Standard means a pure drug, and as well as for sample, it's a market formulations. So after injecting this standard and sample, we have to calculate the percentage purity. Allopurinol label claim is 300 mg, Lesinurad label claim is 200 mg. The percentage assay is 99.93 for allopurinol, 99.95 for Lesinurad. So system suitability, we have to validate the developed method that should pass the system suitability. What are the system suitability parameter? Means tailing factor, theoretical plate resolutions. The tailing factor should not be more than 2. Number of theoretical plates, it should not be less than 2000, and resolution should not be less than 2. So we have to inject this standard solution 6 times, and we have to record the tailing factor value, theoretical plates, resolution. Next, that specificity. So we have to inject the blank. Blank means nothing, but we have to inject only the mobile phase. Then we have to inject the allopurinol standard and as well as allopurinol Lesinurad standards. And we have to inject the standard which contains allopurinol and Lesinurad combinations. So after this, we have to add placebo, and we have to inject that standard plus placebo, and so it indicates that there is no interference due to placebo. Precision. So we have to weigh 300 mg of allopurinol and 200 mg of Lesinurad in 100 ml flask, and we have to add 70 ml, and we have to make a solvent. Further, we put out 0.75 ml into the 10 ml. That gives stock solution of allopurinol and Lesinurad, and we have to inject that sample 6 times. We have to, so we got 6 grams for this precision. 3, 4, 5, 6. So these are the value. It's a peak area values. Peak area means total height in width of. So the software itself will calculate and will give the value. And for this allopurinol, we will get 6 values from the peak area, and Lesinurad we will get 6 peak areas. We have to calculate average standard deviation. The percentage RST we have to calculate. Percentage RST is equal to standard deviation by mean into 100. So so the value should be less than 2. So as per ICG guidelines, the precision was passed with this analytical method. Next, we have to go for linearity studies. The method is linear. The response versus concentration, how long it will be linear. So we have to prepare the stock solution. From that stock solution, we have to pipette out 0.25, 0.75, 1 ml, 1.5, and dilute up to 10 ml with the diluent. And we have to inject that all concentrations of linearity solutions. So the allopurinol linearity concentration solutions are 75, 150, 225, 300, and 375. For Lesinurad concentration 50, 100, 150, 200, 250. So we have to note down the peak area for these linearity solutions. We have to plot the curve, x-axis concentration, y-axis area. So we have to calculate that correlation coefficient. The correlation coefficient should be 0.999. So we got allopurinol, the correlation coefficient is 0.999. As well as Lesinurad, we got the correlation coefficient is 0.999. So it was acceptable limit within the acceptance criteria as per ICG guidelines. So this is a correlation coefficient value which was present within the acceptance criteria. Next, we have to go for how much this method was accurate. So allopurinol and Lesinurad, we have to prepare the stock solution, and we have to prepare 50%, 100%, 150% with solutions which was packed with placebo, and we have to calculate the percentage recovery. This is a chromatogram for 50%, and this is a chromatogram for 100%. This is chromatogram for 150%, and we have to record this peak area. And these are the amount added. 150, it's a 50%, 300 is a 100%, 450 is the 150%. So for allopurinol, the amount found after the study 150.2, 297.42, 447.25. and we have to calculate the percentage recovery. For 50% we got 100.28, 99.14 and 99.39 for 150%. For these three we have to calculate the mean recovery. The mean recovery is 99.60. The acceptable limit is 98 to 102% weight by volume. Accuracy data for Lesinurad, same like that we have to carry out 50, 100, 150. If it is, these are the areas which was recorded. The amount added is 100 milligram, 200, 300 for 50, 100, 150. These are the amount found, and we have to calculate the percentage recovery. So the mean recovery area, we have to calculate average for these three. So we got 100.15, which is the acceptance of 98 to 101%. Next, we have to prepare, carry out the ruggedness. Ruggedness means intermediate precision, the different days we performed. So we got to prepare the stock solution of Allopurinol and Lesinurad from the 300 mg of Allopurinol and 200 mg of Lesinurad in 100 ml. From that we have to make up pipette out 0.75 into 10 ml. So we have to inject 6 times. So these are the 6 graphs from the interday precision. So injection 1 to 6, we have to record the peak area for Allopurinol, peak area for Lesinurad. So we have to calculate average standard deviation RST. So the obtained value which is less than 2, so it was acceptable one within the acceptance criteria of ICCRT guidelines. Robustness. How much the method is robust, it is determined by robustness study. By changing the flow rate, or by slightly changing the mobile phase composition, or changing the column moment temperature, or deduction wavelength, like that. So I changed the flow rate 1.35 to 1.65 ml per minute. The standard solution 225 Allopurinol, 150 Lesinurad used for these studies. So this is a chromatogram showing less flow. If you are decreasing the flow rate, the retention time will be more. If you are increasing the flow rate, the retention time will be less. So even though change the flow rate, the method is robust or not, it is found out by these robustness studies. Like that, I change the organic composition in the mobile phase also, 10% the buffer and the organic phase composition. So these are the chromatogram obtained from this study. Finally, results for variation in the flow of allopurinol. So here the plate count which is obtained from different flow rates, tailing factor which is obtained from the different flow rates. So this is an acceptable limit, because we know that the theoretical plate count, it should not be less than 200, and tailing factor should be less than 2, should not be less than 2. So which was present, it should not be more than 2. So which was present within the acceptance criteria. So like that, we have to go for the Lesinurad variation in flow also. These are the results obtained. Whatever the plate count, resolution, tailing factor, which was within the acceptance criteria as per ICCRT guidelines. Next, we have to calculate LOD and LOQ. So LOD means limit of detection, LOQ means limit of quantification. It's calculated by using the formula 3.3 standard deviation of the response divided by slope of the calibration curve. LOQ is calculated by 10 into standard deviation of the response, slope of the calibration curve. Standard deviation value we can take from the precision, slope value we can take from the linearity studies. So finally, this is we have to prepare as per this method 0.9 gram of microgram per ml allopurinol, and 0.2. We have to prepare 0.09 microgram per ml for solution for allopurinol, and 0.27 microgram per ml of this for LOQ studies. For Lesinurad, we got to prepare 0.06 LOD, LOQ is 0.18. Normally, LOQ is triple of LOD. So we have to inject that LOD sample and LOQ solutions. We got this kind of problems. So results of LOD, signal noise is better one. So this method is able to detect least quantity, as well as it is able to quantify least quantity also. So degradation studies, as per ICCRT guidelines, we have to carry out degradation studies, and we have to prepare the stock as per the normal protocol 300 mg of allopurinol, 150 mg of Lesinurad in 100 ml. From that we got to put out 7 ml and make up to 10 ml. And we go to carry out hydrolytic degradation studies under acidic condition, under alkaline condition, oxidative degradation, thermal induced degradation, like that and all. So these are the chromatogram obtained from the acid degradation, base degradation, and the peroxide degradation, and thermal degradation, and photolytic degradations. So after that degradation studies, these are the peak area which is obtained for allopurinol. These are the peak areas obtained for Lesinurad. So we have to calculate the percentage degradation. For acidic conditions, allopurinol 4.03, for basic 2.53, for peroxide 5.49, for thermal 3.55, for photolytic 5.94. For Lesinurad, for acidic condition 4.06, for basic 3.33, for peroxide 8.31, for thermal 6.76, for photolytic 6.14. So from that we can, as per ICH guideline, up to 10% is acceptable one. So all the degradation values which is less than 2. So this allopurinol and Lesinurad which is stable, all those conditions. So the summary and conclusion: the estimation of allopurinol and Lesinurad was done by RP-HPLC. The assay of allopurinol and Lesinurad was performed with tablets, and the percentage assay was found to be 99.93 and 99.98, which shows that method is useful for protein analysis. The linearity of allopurinol and Lesinurad was found to be linear with the correlation coefficient of 0.999 and 0.999, which shows the method is capable of producing good sensitivity. The acceptance criteria of precision is RSD should not be more than 2, and the method shows precision 0.6 and 0.8 for allopurinol and Lesinurad, which shows the method is precise. The acceptance criteria of intermediate precision is RSD should be not more than 2, and the method should show precision 0.6 and 0.6 for allopurinol and Lesinurad, which shows the method is repeatable, reproducible when performed different days also. The accuracy limit is percentage recovery should be in the range of 97 to 103. The total recovery was found to be 99.60 and 100.15 for allopurinol and Lesinurad. The validation of the developed method shows the accuracy is well within the limit, which shows the method is capable of showing good accuracy and reproducibility. The acceptance criteria for LOD LOQ is 3 and 10. The LOD LOQ for allopurinol was found to be 3.07 and 10.09, and LOD LOQ for Lesinurad is found to be 2.95 and 9.93. The robustness limit for mobile phase variation, mobile flow rate variation are well within the limit. The percentage degradation results are which shows that the method is having good system suitability and precision under given set of conditions. Thank you.